Okay, so the next speaker is John Nardini from NC State University and also a SAMC fellow. He's going to be talking about learning partial differential equations for individual-based models of biological processes. And for those of you who are coming in late, we're going to use the hand raise feature at the end of the talk if you want to ask questions for the last 5 minutes. So 25 minutes of talk and then 5 minutes of questions. All right, go ahead.
All right, thank you, Kevin, for that introduction. Just a quick note that I actually changed my talk slightly, so I actually won't be talking about individual-based models. But if you have questions after the symposium, feel free to send me an email and I can talk some more about some ongoing work on equation learning for individual-based models.
Oh, I apologize, sorry about that.
It's my fault, I changed the title, so. Okay. So the motivation for my talk today is the understanding that mathematical models can provide us with insight into many biological processes, and in particular, partial differential equation models, or PDEs, can help us better understand spatiotemporal processes. As an example of this, during my PhD dissertation, I was interested in studying scratch assay experiments, in which case we grew a layer of human skin cells inside of a well plate, scratched away half the population, and then watch this remaining cell population migrate into the wound area to imitate the human wound healing process. And my collaborators and I were interested to understand an interesting feature of these human skin cells, which is that they maintain physical connections to their neighbors as they migrate into the wound area. So we wanted to use mathematical modeling to better understand how cells use these physical connections to their neighbors to communicate this message of population-wide migration. Ultimately, we were able to use the following nonlinear diffusion equation model in order to do this. And from our interpretation of this model, we could understand that cells were randomly diffusing into the wound area, but also as the cell migrated into the wound, it would pull on its follower cells to migrate along with it through this nonlinear term to incorporate cell-cell interactions. And by simulating this mathematical model, we were able to very nicely fit experimental data on the wound margin of the cell population over time. So this slide was to emphasize that PDEs can provide us with knowledge of biological processes, but when doing this study, as well as other typical math biological studies, where we fit some mathematical model to experimental data, I always had this question in the back of my mind of how do we actually know that our mathematical model is the correct underlying model to describe this given data set? And this has taken me into the realm of equation learning or data-driven mathematical modeling, where our aim is to, from experimental data, directly infer one or a small number of candidate models that may best describe the given data set that we're working with. And so in this talk, I'll be talking about some methods we've developed over the past few years as a way to directly infer mathematical models from experimental data. In a previous study, we were able to show that we could infer some common models that are used for biological processes in the presence of large amounts of noise in the data. I'll talk more about the details of how we infer these equations later on, but the idea of this previous study was that we could infer these common equations, including the advection diffusion equation with up to 25% noise in the data, or the Fisher-KPP equation with up to 10% noise in the data, and the porous Fisher-KPP equation with up to 10% noise in the data, whereas previous methods in the equation learning field struggled to infer these equations in the presence of large noise levels. But of course, the goal for this method of equation learning is to be able to work with biological data and infer a novel mathematical model. However, before we can do this, we need to address some other biological challenges that we're often faced with when working with biological data. And so we took this work to the Mathematical Neuro-Oncology Lab at the Mayo Clinic in Phoenix, Arizona. And this lab often uses PDE models to diagnose patients for glioblastoma from MRI data, where glioblastoma is a highly invasive and heterogeneous cancer of the brain. And some observations that they had about our previous study is that we were still making some unrealistic assumptions about the data that we were working with, where in particular, we were working with on the order of 300 time samples of our underlying process, whereas they're often faced with using 2 or 3 MRI images to diagnose patients. Furthermore, we had all of our simulations being simulated over one timescale, whereas with medical data, you may have a variety of different timescales over which your data has been measured. And also, we only inferred one parameter value for each one of these equations, but biological processes such as glioblastoma can exhibit a large level of heterogeneity between patients. So in this study, we're again going to use an artificial data simulation as a proof of concept. And in particular, we're going to focus on the Fisher-KPP equation, which models the spread and proliferation of a population over time. And we're choosing this equation because of its wide use in the mathematical biology literature, not just in glioblastoma progression, but also in ecology and wound healing. And we're going to look into the performance of our equation learning methodology in the face of working with a small number of time samples, such as either 3, 5, or 10. And we'll work on 2 separate timescales of either half of a year or 3 years over which our data is measured. And we'll work over a slew of parameters that are relevant for glioblastoma. In particular, for the parameter values that we'll consider, each of these dots here corresponds to estimated rates of diffusion on the horizontal axis, as well as proliferation on the vertical axis that have been estimated previously from glioblastoma patients. And so we see from this figure that both of these parameters can vary over 2 orders of magnitude. So our equation learning methodology should be able to correctly infer equations over this wide variation in parameters. To first capture the mean trend of the population, we're going to focus in on this red rectangle given here as it captures the main trend in the glioblastoma population. And to start off with, we'll focus on the 4 corners of this rectangle, where in the bottom left corner, we have a slow simulation where the rates of diffusion and proliferation are small. And in the simulation of the Fisher-Kolmogorov equation at these parameter values on the right, we see that we start with a small bump in blue and eventually it develops into the magenta curve at the final time point. And then moving rightwards, we can arrive at what we'll call a diffuse simulation with a larger rate of diffusion. And we see that this population at these parameter values will spread out much further than the slow simulation. And then moving to the top left corner, we have what we will call the nodular simulation, which migrates at a similar rate, similar speed into the wound area. It invades at a similar speed as the diffuse simulation, but it maintains a much sharper front. And lastly, we'll have a fast simulation, which also maintains a very sharp profile, but moves at a much faster rate than the previous populations. So with those parameter values that we'll consider, we're again going to use an artificial data simulation study throughout this. So to further explain that, in order to make each one of the data sets that we'll be working with, we'll begin by simulating a partial differential equation model over time, where we assume that the rate of change of the variable u, which pertains to cell density, is equal to diffusion plus logistic growth. And then once we've solved our PDE model using the method of lines, we can then create proportional error. We can approximate noisy spatiotemporal data by adding white noise at each spatiotemporal point. And the statistical model that we're using here assumes that the variance of our data will increase with the magnitude of the solution to the PDE model. The motivation for this proportional error model is because often if we're looking at cell density, we may be counting the number of cells in a given spatiotemporal point, and it's much harder to count 1,000 cells than it may be to count 10 cells. So these are just how we generate the data sets that we'll be working with throughout this study. And then in the third step, we want to work with this noisy spatiotemporal data and uncover that the Fisher-KPP equation is the PDE model underlying this data from many possible candidate models. For this third step, we've developed the following pipeline in order to do this. And in this pipeline, we have 3 main steps, where beforehand, we assume that we've measured or collected some noisy spatiotemporal process. And then in the first step, we perform data denoising and differentiation, where we obtain a smoothened version of the data and then use this smooth inversion to estimate the derivatives of the underlying PDE model. And then once we have these clean versions of U and its derivatives, we'll then use an equation learning methodology to infer a small number of candidate models that may best describe this PDE model. And then finally, we'll use a model selection step to choose which of these three models is the best in describing the data and perform preliminary analysis for the model, including parameter estimation and uncertainty quantification. So in the first step, we're interested in data denoising. And this is work with John Lagergren, who is a graduate student at NC State, and we'll be talking next. So previous methods of equation learning have relied on finite differences for numerical differentiation or polynomial splines for data denoising and then differentiation. But our previous study showed that these methods are very sensitive to the amount of noise in the data. And instead, we'll use a neural network approach for denoising and differentiation, which we found to be much more robust to noise in the data. The motivation for this neural network is that we'll use it as a universal function approximator, meaning that if we have a continuous function on a compact domain, then we'll use this neural network to approximate this continuous function. And the accuracy of our approximation should increase as we increase the depth and height of this neural network, where depth corresponds to the number of layers and the height corresponds to the number of neurons in each layer. And so the basic architecture of this neural network is that we'll have two input values, x and t, to denote a spatiotemporal point, and we'll have one output u to make our prediction for what the PDE solution is at this spatiotemporal point. And then in the neural network, we just use a simple feed-forward neural network with 3 hidden layers and 256 neurons in each layer. And then once we've trained the neural network to match the spatiotemporal data, we can then use backpropagation to estimate derivatives such as ux, uxx, and dudt. And again, in order to train this neural network, we're going to use a loss function comprised of these two terms given here, where in this first term, we're just comparing the output of the neural network directly to our data. This is similar to mean squared error. However, because of our proportional error model, we are normalizing by the output of the neural network. And in the second term in our loss function, we sometimes saw when training the neural network to data from a sparse number of time samples that the neural network would output unrealistic values between time samples. So in order to avoid this, we're going to penalize the neural network for outputting values that fall outside the range of values observed in our data. And while the first loss term will be evaluated wherever the data is sampled, we evaluate the second loss term on a finer grid that covers the entire spatiotemporal domain to avoid unrealistic output values from the neural network. And in our previous study, we were able to show that by training this neural network to noisy spatiotemporal data from the Fisher-Kolmogorov equation, we can obtain very good derivative estimates, where in the top left corner in blue, we have our prediction, our noisy data that arises from the Fisher-Kolmogorov equation. And then in the top right corner, we have our predicted value of u in red from the neural network, as well as a true underlying value of the PDE in black. And then along the bottom row, we have the same predictions for derivatives in red, as well as their true underlying values in black. And we see that these curves are very close to their true underlying values. So we'll use this neural network for data denoising in this methodology. And then once we have these clean estimates, we'll then use this we'll use these in our equation learning methodology to find the small number of candidate terms that best describe this data set. So we're going to use the PDE find algorithm to do this, where PDE find stands for PDE functional identification of the nonlinear dynamics. This algorithm has become popular over the past 3 years, and in the next 2 slides, I'll just talk us through the main ideas behind this algorithm. So to start off with, let's assume that we have data that does satisfy the Fisher-KPP equation, and let's focus in on one spatiotemporal point, Ti xj. Then we know that this data satisfies the following differential equation of the rate of change of u is equal to diffusion plus logistic growth. And we can distribute the lambda u term to arrive at the right-hand side of our equation, which we may notice is a linear combination of terms that pertain to u. So we can rewrite it as as a dot product. And of course, this equation will be true for any value of u. So if we consider the vectorization of u, we could write it as in the following linear system, where we have that the vector of time derivatives is equal to the matrix whose columns correspond to u, u squared, and uxx, being multiplied by the 3×1 vector of parameters. Now, of course, we have no idea if our data actually satisfies the Fisher-Kolmogorov equation or not. So instead, if we have no idea what the true underlying equation is for our data, we may instead build a library of many potential right-hand side terms for the PDE model. And we'll call that large library of potential terms theta. And if we're focused on reaction diffusion equations, then we may include polynomials, derivatives, and cross terms between polynomials and derivatives in this library theta. And for simplicity in this study, we'll keep p fixed at 2. And then once we've built up this library of potential right-hand side terms from the neural network, we can then perform sparse regression on the linear system given by du dt is equal to theta xi, where the only unknown here is xi, which parameterizes the right-hand side of our equation. Now, of course, we don't want to have a PDE model with 10 terms on the right-hand side because that may not be very interpretable. So we'll use methods from sparse regression to ensure that there's only a small number of terms on the right-hand side, and thus the inferred model is interpretable. Also, in our previous study, we were able to show that the PDE find algorithm is sensitive to the training and validation splits over which it's trained over. So for example, we'll use a 50-50 training and validation split of our spatiotemporal domain for inference. And here's just a quick example where over 5 different training and validation splits, our inferred equations are the logistic equation 3 times, the Fisher KPP equation 1 time, as well as this other equation, the final time. So to account for this uncertainty, what we'll do is we'll count the 3 equation forms that are most commonly recovered, given here, and then we'll perform a final round of model selection by simulating these 3 models and starting from an initial condition given by the initial time points. And then we'll compare these top 3 models to the final data time points using an AIC score, where we want our final model to match the data well, but it will also be penalized for having a large number of parameters. So in this figure here, we have the logistic equation in purple, which was the most commonly recovered form, but we see that the simulated model does not fit the experimental data very well. Whereas the second most common form was the Fisher KPP equation in yellow, which fits the data very nicely. And then the third most frequently recovered form in green also does not fit the data as well. So in this example here, the Fisher KPP equation in yellow would be the final selected model form. So with that, we can now start talking about some results of our equation learning methodology over a range of parameter values that are pertinent to glioblastoma. And we'll start off with results that hold for the slow and diffuse simulations. So I'm going to start off by discussing the diffuse simulation, but the results are also pertinent for the slow simulation. So to start off with, we're going to look at our performance in inferring this correct equation given in the top over a short time interval of half of a year. And with only 3 time samples of the data, we observed that the logistic equation is inferred and the parameter estimates look pretty bad. Next, we could look at the inferred equation with 5 time samples, and we still get an incorrectly inferred equation. And with 10 time samples, we again still get something that doesn't match the true underlying mathematical model. But we notice that this appears to be the linearized form of the true underlying model. And this inferred equation with 10 time samples makes perfect sense when we look at the data over which we were training over, because we see that the dynamics of this model haven't changed too much from the initial condition over this short time interval of half of a year. So it makes perfect sense that we're recovering the linearized form of the true underlying model. If we simulate the model over a longer time interval of 0 to 3 years, we see that for all cases, we infer the correct underlying form, as indicated by the green shading of these cells. And as we increase the number of time samples, our parameter estimates tend to increase. And also when we observe the underlying dynamics over this longer time interval, we now see that we are able to observe both the linear and nonlinear dynamics of the system, where we have an initial growth phase, but also start to approach the carrying capacity of 1 for the model. Next, we can talk about some results that are pertinent for the nodular and fast simulations given on the top of our rectangle. And here the opposite results hold for this faster simulation, where over the shorter time interval of 0 to half of a year, we're able to infer the correct underlying equation on the shorter time interval with 5 or 10 time samples. And if you look at the dynamics over this shorter time interval, we again see this healthy combination of both linear and nonlinear dynamics. Whereas on the longer time interval of 3 years, we see that we were never able to infer the correct underlying equation. And if you look at the dynamics of the system, we observe that between the first and second time points in blue and green, the dynamics have already converged on their traveling wave form. And this is an issue for our methodology because traveling waves are not identifiable since many reaction diffusion equations may give rise to them. So with that, and with these results, we were able to show that when we consider the simulations with lower rates of proliferation, then we can infer the correct underlying equation with as few as 3 time points over a longer time interval of 0 to 3 years. And then when we consider the simulations with larger growth rates, we can infer the correct underlying equation with as few as 5 time points over a shorter time interval of half of a year. Now, these results aren't terribly surprising because if we were to analyze this equation in a typical mathematical modeling approach, the first thing we would probably do is non-dimensionalize time by multiplying time by this value r. But it's good to know, it's good to have these solid results, as well as to understand that a informative data set for our methodology should exhibit both linear and nonlinear dynamics. Next, we can also focus in on a finer grid of diffusion and proliferation values, where here on the left and right, we have 5 by 5 grids of diffusion and proliferation values. And our 4 previously considered simulations are given in the corners of these grids. And in these grids, we ran our equation learning methodology for data sets generated at these parameter values. And white cells correspond to data sets where we were able to infer the correct underlying form, and gray cells are where we were not able to infer the correct underlying form. So on the short time interval of half of a year, we see that we tend to be able to infer the correct equation for larger rates of proliferation, such as between 5 and 30. But with a lower growth rate of 3, we start to have more and more trouble inferring the correct underlying equation. And similarly, over the longer time interval of 3 years on the right, we can infer the correct equation with smaller rates of proliferation, such as 3 and 5.3. But then as we increase the rate of proliferation, we start to have more and more issues inferring the correct underlying equation. And furthermore, it's not just enough to be able to infer the correct equation form. We also want to ensure that when we train this methodology to infer models over the appropriate time intervals, they should be used to predict unobserved dynamics. So here's an example where we trained the slow simulation on its inappropriate time interval of half of a year, and we see that the predicted model fits the data that it was fit to very well over this time interval of half of a year. But then when we simulate this model over the longer time interval of 3 years, it does not generalize very well and suggests a carrying capacity that's 5 times lower than the true carrying capacity. If instead, however, we perform the same prediction by first training the slow simulation on its appropriate time interval of 3 years, then we can use this correctly specified model to predict the dynamics over the shorter time interval of half of a year. And similarly, the same results hold for the fast simulation, where when we train it on the correct time interval of half of a year, it can then generalize well to predict the underlying dynamics in the right over the longer time interval of 3 years. So in conclusion, we've introduced a new equation learning methodology that's comprised of 3 main steps including data denoising, equation learning, and model selection. And we showed that in order to infer the correct underlying model, we need to sample the data over an appropriate time interval. And this time interval should exhibit both linear and nonlinear dynamics of the system for correct inference, as well as prediction for unobserved dynamics. And we focused in on glioblastoma as a test case in this study. However, the wide use of the Fisher KPP in the mathematical biology literature will make this study even more applicable to a wide range of biological systems. Some future work for this project will include the ability to automatically detect what high and low content data sets may look like without having to actually run the equation learning methodology, as well as exploring the results that occur for other common math biological models that may be used in the literature. So thank you all for listening, and I have too many collaborators to thank by person for this project, but it's all of these individuals listed here from NC State, the Mayo Clinic, the University of California, Merced, as well as the University of Nottingham. And on the chat later on, I'll post links to our previous paper and preprint.